Hello, folks. I'm Dr. Subroto. Along with Dr. Aman, we are going to share some of the knowledge about human body and physiology today with you. In the first segment, we will be talking about the control and developmental processes in various structures like the brain, the nervous system, reproductive, skeletal, and so on and so forth. Let's begin with the cells. We have trillions of cells, and they together constitute what we call tissues. And similar kind of tissues, they form organs, and these similar kind of organs will form altogether a organ system to do a particular function. There are various systems in human body: nervous, endocrine, reproductive, sensory, muscular, skeletal, circulatory, the respiratory, digestive, the immune system which fights diseases, and ultimately the excretory system. Now, to begin with, what is homeostasis? Obviously, this is a question which is very important. Our body tends to be at balance and in stable conditions with relation to the external and internal environment. And for example, if the temperature increases, we start sweating so as to cool our body. At the same very time, when it's cold, we shiver so that the muscles they contract and generate heat. So this type of balancing the temperature with relation to the outside vis-a-vis the inside is referred to as homeostasis, that is making a equal status or a balanced status. Similarly, oxygen level in the blood is going to maintain is going to be maintained within the blood vessels by taking oxygen whenever we have less of it and by removing carbon dioxide. We have water balance too. Again, the kidney does the function, and you always know that you always try to balance the pH, the acidity and alkalinity of the blood, which is very essential. So when our blood becomes acidic, we do take more alkaline food to balance it. Of course, this is also done by various salts and water. Let's begin with the nervous system. One of the most tricky, important, interesting one. Consists of two parts. The central, mainly consisting the brain and the spinal cord, and the peripheral nervous system consisting other parts of the nerves, other nerves of the body. Let me give this privilege to Dr. Aman, please.
So as you just now mentioned, Dr. Subroto, that we have one system which runs in the central axis of our body, central nervous system, and the nerves coming from it runs in the periphery, which is called peripheral nervous system.
True.
The central nervous system is again enclosed inside our skeletal system. The skull encloses the brain, and the vertebral column encloses the second part, that is our spinal cord. And the peripheral nervous system, the set of nerves which go to the body parts, which can be controlled by our own will, that is voluntary, we refer to as somatic nervous system.
All right.
And there is another part which
Autonomic.
which is involuntary. For example, the involuntary muscle, for example, the heartbeat, for example, rate of breathing.
So we don't have a control over it.
We don't have control over it. So this is called autonomic nervous system.
It
And
can
again,
be
it has two parts.
sympathetic, parasympathetic.
Because there is one which functions when we are under normal conditions, that is parasympathetic. But suppose body goes under some stress, say fear, pain, anger, any such situation,
That would be
then
sympathetic
the
system.
sympathetic nervous system will start working.
So this is the nervous system, as you are able to see. The central nervous system comprises the brain and the spinal cord. The rest of it has been explained in the previous slide. Let's go to the basics of a brain in a nutshell. So we have about 1.5 kgs of brain. That's a lot of it. What do you think, Aman? And very, very complex one. And we can see lots of depressions and swellings like waves. What are these?
Well, this is to increase the surface area.
Are they called by some names?
Exactly. The one which are the grooves, we could call them sulci, and the one which is a raised area, that is gyri.
Gyri and
Yeah,
sulci.
and this is a feature which belongs only to the mammalian. Now in brain basic, we can divide brain into three parts: the forebrain, where we have cerebrum, the midbrain, and the hindbrain. Hindbrain, which comprises the brainstem and cerebellum, it's the vital part, and brainstem has mainly the part medulla, which controls the breathing and respiration.
Cerebrum happens to be the largest part of the brain, which takes lots of information, processes them.
It's 9
And
o'clock.
it is split into two parts. And each of these are referred to as hemispheres, the right and the left. In fact, the left hemisphere controls the right side of our body, and the right hemisphere controls the left side of.
So you can see here that each of the hemisphere, it controls its own respective part. The one part will be mainly the cognitive or the memory section, and another one will be controlling the muscles.
The question is, which hemisphere controls the cognitive functions?
Well, it is dominated either by right or the left cerebral hemisphere. In fact, it depends upon whether the person is a right-handed or the left-handed.
Suppose I am right-handed.
Then in your case, it will be the right hand which will be cognitive, because the left hand means it is controlling your muscular activity or the motor activity.
So these are the two, right and the left hemispheres, and they deal with specific work. For example, the left hemisphere would take care of mathematical solutions that one might give, the reasoning one has, the language. Are you good at language? I am not. What about you?
Well, it stores the different languages.
So your left hemisphere would be better. At least it can do a better work in terms of the language.
Yeah, whenever we learn the new languages, they are stored there.
Okay. And detail orientations. For example, the right hemisphere, however, takes care of your emotions. It governs the musical sense that you have, isn't it?
Yeah. So this is how, the right hand side of our brain controls the left, and left hemisphere controls the right side of the brain, and the left mainly is for the linear thinking kind of a mode, and the right one is mainly controlling the holistic thinking. And again, our cerebrum, that is our forebrain, it is well differentiated into the four lobes. The one which is in the forehead is the frontal lobe. On the top is parietal. Then we have occipital, totally in the last edge of the brain, and the temporal region, which controls the sound and our languages. So this is the frontal, and then we have the motor and the cortex area in our near the parietal lobes, and occipital, which is in the blue color. It is the seat of our vision. Temporal in yellow color is mainly for the hearing processes or the auditory sense.
So here's the brain. The front portion referred to as the frontal lobe. It helps in organizing everything, right? And the memory.
We can say it is a seat of reason and our
speech.
Thinking or speech. Yeah.
Wow. Then you have somewhere on the top, on the roof, we have the parietal lobe.
Mainly it is a center of sensations, all the touch, heat, cold, pain, whatever we feel, that is associated with this area.
Okay. Then we have the temporal lobe near the ears. What does this one do?
Well, again, as I mentioned already, that this is the seat of our sound, or even producing or understanding the speech. And many aspects of memory are also associated with it. So this is the seat of the vision, occipital lobe.
Yeah. Oh yes, I think this one governs the balance of your body.
So it is very well developed
Cerebellum.
in the gymnasts, because while swimming, while walking, even when we are going upstairs or downstairs, our movements of our feet and legs, this is under control of cerebellum. In fact, the whole coordinated movement of the muscles in our body are under control of this part, cerebellum.
So, Aman, if I give you 3 pegs of wine, I think you would lose all the coordinations.
Exactly.
So would you be able to drive?
No, we can't, because alcohol affects this part of the brain,
the cerebellum.
cerebellum.
This slide shows the brainstem.
Well, this is a vital part of the brain. Any small injury to this part can cause instantaneous death. Reason being, this is a portion of the brain which is controlling our vital function, say our heartbeat, our rate of respiration, even our
Breathing.
digestion.
Swallowing.
Yeah, they are all associated with this part of the brain. So
Very
you must have
sensitive.
heard, sometimes that a person, just by minor slip, say in the bathroom or even on the stair, instantaneously die.
Yep.
So the reason is the damage to this region, especially the medulla part.
And that's where you would see the first thing that a patient is taken care of in an accident is that they would put a collar. They would put a collar around his neck and
Yeah,
then put him
because
on the stretcher.
exactly, that is the part which has to be protected, because if anything goes wrong with cerebrum, we may lose memory or speech. Anything goes wrong with our say the midbrain, we may lose vision. But if something goes wrong with this part, instantaneous death. So this brain stem is very, very essential.
It controls the heartbeat,
Yeah,
the
so you see
respiration,
all the
the
three,
digestion,
all the
that is
vital,
swallowing.
all the vital parts of the body.
Okay, we have another structure called thalamus. Can you tell something about the thalamus, which is right in the center of the
Well,
brain?
in a simple terminologies, I'll call it a gateway.
Mhm.
It is gateway of the body to brain and brain to body.
And at the floor of it, we have the hypothalamus.
Hypothalamus is the one which is having the master gland attached to it, hypophyseal gland, or what we mentioned to as pituitary. So it controls the emotion, even our hunger, thirst, our body temperature, our whole biorhythm, circadian rhythms. They are controlled by this part, hypothalamus.
Now, I often see you getting up early in the morning, say at 3:30 or 4, and you hardly make a mistake without your alarm. How is that? Is it because...
Well, again, we call it circadian rhythm. It's that it records the timing.
Okay, so your hypothalamus might be working very well.
For anyone, anyone, if they sleep at especially one particular, say one person sleeps at 10 o'clock. So even without looking at a clock, one will start feeling sleepy at 10 o'clock because of the circadian rhythm in hypothalamus.
Mm-hmm. Question: what part of your brain allows you to consciously hold your breath?
Now again, see, it's the word is consciously hold your breath. That means it is voluntary. It is under our control. That means we are talking about the cerebrum.
Okay.
But in the second case,
what part makes you finally take a breath?
now, while taking a breath, well, this may not be in our control. It is something involuntary, because it is something
automatic system.
The muscles which have to work at their own will. That means we are talking about cerebellum.
So that is the cerebrum and the midbrain that we talked about. Well, coming to cell types in the nervous system, two kinds: neurons and the glial cells. Obviously, one understands that nerves transmit electrical impulses, which are referred to as neurons. The others, which support these cells and protect them and insulate them, are referred to as glial cells.
Well, you know what I will say, that neuron can be compared to electric wire, and the glial cell can be compared to the plastic insulation over it.
Wow. Let's see a typical neuron.
Well, we will say neuron is a fundamental component of our nervous system.
Two parts. If you can see a cellular part, fine blue in color.
So we will say it is having cell body and it is having cell processes. The cell body is the one which has nucleus, and the cell processes are dendrites, the one which always receive the stimuli from, say, another neuron, and the one which takes away the impulses from this neuron are the excites, which are attached to a part called as axon.
Oh, so this axon is nothing but the tail-like process that we are able to see here.
Yeah, it always takes messages away from the neuron to another cell.
And I can see a myelin sheath insulating it altogether.
Well, it is made up of some special kind of lipid called a cerebrolipid, which do not allow the ions to pass through.
Okay.
So it's a covering again. We will say it is a modified glial cell.
So here is the cell again, the nerve cell attached to a muscle cell, and the terminal arborizations are just joining the motor end plates of the muscle.
Which I mentioned as excites,
Mhm.
because they are the one which are taking the impulses from this blue colored structure, that is neuron, to the effector, that is the muscle.
Now, many people do ask, what is the direction of the impulse? Is it from the cell body to the axon, or from the axon to the cell body? Now let me tell you beforehand, the cell body has got these fine pink projections called dendrites, and the axon leads to small bulb-like terminal ends. Am I correct?
Exactly.
And each of these terminal ends are attached to the dendrites of another cell.
Because each of the terminal bulb is actually storing a chemical which is referred to as neurotransmitter. We will talk about that little later while transmission.
So what is the direction?
The direction will be dendrite to the body cell, from body cell to the axon, from axon to excite, that is terminal button, which will be connected to another neuron through the chemical messenger.
That's being shown here now.
Yeah.
Well, there are three categories depending on the origin and destination of the message. The three kinds are sensory, the motor neurons, nerve cells that is, and the interneurons.
So all those cells which are taking messages, say, towards our central nervous system, we call them sensory. And all those which pick up messages from the central nervous system to our effectors. Effectors means our muscles and glands. They are referred to as motor cells. And suppose there is some cell which has capacity to take the message towards the brain and spinal cord, and also bring the message from brain and spinal cord back to the effector. Such mixed type of cells are referred to as interneurons.
Oh, they are in between.
That is, all the cells of spinal cord are interneurons.
Two types of motor neurons. Somatic, nervous system controls the voluntary actions. This we know, this section we know. And the other section is autonomic nervous system, which works automatically.
So all those motor cells which were actually taking messages to our muscles and glands, so those ones, they are, which are under our control, they are somatic, and the one which are involuntary, we will call them autonomic motor neurons.
The same is being shown here, how they are interlinked to each other. As you already told, the autonomic nervous system has got two sections, sympathetic and parasympathetic. Sympathetic system works at the time of stress.
We will say only emergency, whenever
Stress.
body has some emergency.
And why is it called fight or flight response?
Because depending upon a situation, an organism may like to fight or may like to flight from that situation. So someone is scared of a snake, so someone is brave enough may kill the snake or may fight the bull as shown here, and another person may run away from it. So you can either fight or you can flight. So parasympathetic is when we are relaxing, and both these systems are antagonistic to each other. That is, both of them, they control the similar organs. But sympathetic nervous system, whenever we are in stress, even if we are hungry, the digestion processes cannot occur because they suppress our digestive glands, where in parasympathetic they help in digestion.
How neurons work. Now, nerves, they use some kind of electrical signals, as we know. And the electrical signals can only be sent if there are differences in the voltage. obviously across the membrane. So same way as the electricity works, we have what we call the potential difference. In nerve cells, we have electric potential across the membrane, which we refer as membrane potentials.
So this shows there will be differences of the ion concentration outside and inside the cell. And
You mean outside and inside the neurons or the membranes?
outside the inside of the neurolemma, that is the
That is
membrane
the
of
membrane
the
of the nerve cell.
nerve cell. So outside generally it is the sodium which is dominant along with little bit of chloride ions, whereas the inside is having many kind of proteins, amino acid, along with that carboxyl ions are abundant and potassium ions are abundant. So the interior in comparison to outside is slightly negatively charged.
So outside positive, inside negative.
And outside dominantly sodium, inside dominantly potassium.
And whenever the cell is not doing any function or not sending any signals, it would have a potential difference, and that would be referred to as a resting potential. And we find that it is something somewhere equivalent to minus 70 millivolts.
And it will be maintained because the sodium which is outside cannot enter inside because the gates are closed when the nerve is resting.
That is at resting potential.
And similarly, potassium cannot come out because potassium gates are also closed when our nerve is resting. So this potential difference of minus 70 millivolt is always maintained.
In this slide, we are able to see what's inside and outside the cell membrane.
So when the cell is at rest, we can see the outside is positively charged, but when the action potential sets in, or when we send some stimulus to it, then the positive ion, that is sodium, start moving inside. The gates open for sodium, and action potential sets in.
Yeah, and I see the positive ions getting inside.
Yeah, that's the reason that from minus 70 millivolt, the action potential becomes 0 or maximum to plus 30 millivolt.
So in order to set what you call nerve transmission, we need to give a stimulus first.
Exactly, and the stimulus has to be all, not little bit. Suppose you are sleeping and I tickle under your feet. So if it is not good enough, you may just remove your feet and then again go to sleep. But if it is powerful, then you are going to get up and react. What are you doing?
So you mean it should be all or none?
Yeah, and it has to be all or none, because if it was little stimulus, then all must be roaming naked. We can't wear the clothes because clothes will be irritating the skin. But the little stimulus does no effect to the body.
So you mean there is a minimum amount of stimulus called threshold stimulus?
And that's why it is called all or none principle.
Here is an action potential.
So as we were talking about that, when the nerve is resting, the sodium gates are closed, and even potassium gates are also closed. That means there are two kinds of gates in our membrane.
For the potassium and for the sodium.
Yeah, which allow the ions to move in. So when we give any stimulus, the sodium gates open first. So what we have to remember here, what is most important here in this slide, that sodium gates are first to open, first to close. Okay. Potassium gates always open slowly, and similarly they close also slowly.
They are the second ones.
They are the second one. When the action potential is peak, say plus 30 millivolt, then that is the time when sodium gates will close down fully. And that is the time when potassium gates will start opening, when the action potential sets in.
So that just below the dotted line, we have the resting potential. Yeah. And a little more stimulus will allow the threshold level to be accomplished. Yeah. And then the action potential sets in.
So once action potential sets in, the potassium gates now open and potassium starts moving outside.
We'll be talking about this a little
later. So here is a minus 70 millivolt. The sodium gates were closed, and once we give some stimulus, the sodium gates open. sodium start flowing inside. So whole of the sodium has come in. When whole of the sodium goes inside, then this is the time the action potential sets in.
Okay. So this is what you're trying to show, how the action
So it is a
potential sets in.
minus 70 millivolt in the beginning. Then we give stimulus, the gates open, whole of sodium has gone inside. So the gate will close down now, inactive. That means action potential is 0 or plus 30 millivolts. So this in the resting state, outside is positive and the inside is negative. So once the potassium, it has all gone out and the sodium is inside. Now when we have to bring this nerve back to the normal state, back to minus 70 millivolt, this is the time which is called repolarization, and this requires energy. So there is one sodium
returning of
back to normal.
Okay.
So there is one enzyme which is ATP mediated, the carrier protein, and this carrier protein has a capacity to attach 3 sodium and 2 potassium at a time.
So it provides the potassium.
So it swivels, this enzyme swivels and it attaches sodium and then swivels and throws the sodium out, and the same very time attach 2 potassium. That means this ratio is very, very important for us to remember. 3:2.
Okay, what I get that we throw out 3
sodium for
ions,
2 potassium,
and we get in 2
potassium. And that's the reason I mentioned in the beginning that sodium, it moves faster inside, faster outside, whereas potassium, it moves slowly outside and it comes very slowly inside. So there must be one stage during repolarization when the whole sodium has gone out but little bit potassium is still left outside.
True.
This state is referred to as hyperpolarization.
All right. What are you trying to show here? Potential difference occurring on this slide, for example. I can see the threshold being given onto the left hand side of the screen.
Yeah, so see here we say potassium efflux. So this red one marking is hyperpolarization. So this is a state when whole of sodium has gone out, but you can see here one red dot, one potassium is still left out. So till whole potassium goes inside, we cannot give another stimulus to this nerve. So therefore this state is referred to as hyperpolarization.
All right. Can we do it one more time? We can start from the threshold. Yes.
So it is minus 70 millivolt. Now the
Okay.
sodium gates will open because we have already given the
There
stimulus.
it opens.
It opens, and now the action potential sets in. It becomes plus 30 millivolt. That means sodium gates are closed. Potassium are now open. This red marking shows potassium is open. So see, the potassium has started going out. This purple one. These are the potassium gates.
I can see that.
Yeah. So here whole of sodium has come in and the potassium goes out.
So this peak continues
This
and continues to inflow.
Yeah.
Okay. And thus the sodium keeps going out and the potassium keeps going in.
But
and
once
keep
we have
changing.
to throw them out, every time we need ATP molecule because the repolarization is energy mediated process.
So you mean for this action we always require you explain all or none principle, or the phenomena. That is, you give everything.
You give either full stimulus, the little stimulus is not going to influence the body. Otherwise, we can't wear clothes because they will irritate the skin. You must have seen sometimes there is some patch inside attached to our, say, the garment which we are wearing in, and it irritates our skin.
Yes, it does.
Yeah, because it is giving us a lot of stimulus, which the other clothing was not given.
Okay.
So generally many people are allergic, so they have to remove that batch from inside.
Okay.
So that is all or none principle.
So you give full or you don't give any. Let's explain a little more.
So this slide depicts that how action potential propagates from one end to another all across the membrane of the axon. So in between, in the internodes we have myelin sheath which doesn't allow the ions to move through. The ions only move from the nodes of the Renway. So there is a benefit here, that is the message will be jumping from node to node, thereby increasing the rate of the transmission.
You mean in the node of Ranvier, it is not insulated?
And so the sodium can come in and potassium can go out.
Okay.
So again there is one period when another message cannot be taken by the nerve. That is a stage called as refractory period or the hyperpolarization state, because whole of the sodium has to go out and whole of the potassium has to come in case we want to give another stimulus.
Is that the reason why it prevents the action potential to be going backward?
Exactly.
Now, this is the action potential graph.
So we already discussed this, that the sodium gates, they open earlier and they close also earlier. So once the resting potential was minus 70 millivolt and the action potential sets in, once it reaches the 0 and then plus 30 millivolt, this is a stage when sodium gate will close down and potassium will open.
So when more sodium channels open, the peak goes to the maximum. Shoots. And
once it is on the maximum, now the potassium gates open and the sodium closed down.
So the peak goes down.
So the potassium starts going out. So there is one stage when the whole of the sodium during repolarization has been taken out, but still little bit potassium is left out. That stage is referred to as hyperpolarization.
Do I see that in the bluish region?
In the bluish region and down where the peak is bending down.
Yes.
So that is about minus 40 to minus 50 millivolt.
Okay.
That will be the hyperpolarization region.
Even lesser than
the resting potential.
Minus
so once
70
the whole
millivolts.
potassium goes in, then only it will be back to normal to minus 70 millivolts.
Only then the next resting
so
potential
during
sets.
hyperpolarization, new message cannot. So this is again explaining the same phenomena. So as I was mentioning, that myelin sheath, it is very helpful from allowing controlling the speed of the movement of the signal, because the signal will be jumping from node to node. This is called as saltatory conduction.
This saltatory conduction is one of the phenomena how the electrical transmissions occur.
And that's why vertebrates are always faster in their response in comparison to invertebrates.
Because they have membranes?
They do not have myelin sheath, the invertebrates.
And therefore it is slow in their case.
Therefore the message is very, very slow in their case.
So here we are seeing how the action potentials keep moving from one nerve to another.
From one node to node, see it's jumping.
Yes, from node to node and also from
one neuron to another. That is dendrite to axon, dendrite to cell body to axon, and again axon to excite to again dendrite of the next neuron.
Isn't it a beautiful slide? Just like, of course, go ahead.
So from excite of a neuron to the dendrite of, say, the B neuron, there is always a space of 100 angstrom that is referred to as synapse.
So synapse is a junction between two nerves.
Junction between two neurons.
Yes, that's true. So what happens in a multiple sclerosis?
Well, there is degeneration of the myelin sheath. So you can imagine if there is no myelin sheath, then the message will be very, very slow, and sometimes even the person becomes totally paralyzed in the worst of the condition.
Myelin sheath is the case. So how does these neurons they work? Via the electrical junctions in the gap junctions. That is, in between the two cells, there are certain gaps.
Yeah.
How do they work there?
Well, in the case of the heart muscles, the message goes through the gap junction.
Okay, and
that's why we call it electrical stimulus in their case.
Okay, what are neurotransmitters?
So when there are no gap junctions and the movement occurs slowly, we have the chemical synapses, that is one neuron to another neuron. Instead of the gaps which were there in the heart muscles, here it is the chemicals which will be carrying the signal from A neuron to the B neuron. And
that is at the synapse.
That is at the synapse, and these chemicals are stored in the synaptic bulbs or the terminal dendrite excites of the axon, and this is how, once the action potential reaches those bulbs, the neurotransmitter will be released in the gap and it will get attached to the dendrites of the B neuron.
Let's have a look at it. Here it is. Okay, on the left you have a cell body, that is a nerve cell, which is trying to tell us that yes, these
It's 4 o'clock.
Okay, it's 4 o'clock. There is synthesis of neurotransmitters and they pass down and come to the terminal ends or the bulbs. Now, they are released here.
So once the
These
action
neurotransmitters.
potential reaches this bulb, which is storing the neurotransmitter,
Mhm.
they will get attached to the membrane. So we will say synapse has three components.
Mhm.
We have the presynaptic cell
Mhm.
or the membrane. Then we have the gap, which is called as synapse. Then the second neuron will be referred to as postsynaptic neuron.
That is shown here as orange color.
Yeah, that is whose dendrites are going to receive that chemical.
Okay.
So a neuron, that is presynaptic neuron, is going to release the chemical into
As you are able to
synaptic
see the
junction.
purple colored dots being
Yeah,
released.
that is, they are the neurotransmitter, very common, acetylcholine,
Mhm.
and they'll come and attach to the chemoreceptor site of the dendrites.
I am able to see some of the receptors on the cell surfaces.
That is of the postsynaptic neuron.
Mhm.
And once the vesicles are empty, they will re-enter back to their this presynaptic bulb, and this whole release process is controlled.
So what happens when neurotransmitters diffuse to the target cells, which you explained sometimes
So
ago?
once it goes to the target cell, which we were calling postsynaptic cell,
Mhm.
the attachment of the neurotransmitter will send a chemical signal to that membrane. Thereby, that chemical will
Triggering.
help to trigger the stimulus and open the sodium gates. And then the same process will occur, which we learned previously.
The action
That is the
potential.
resting potential minus 70 millivolt, changing to action potential, that is plus 30 millivolt, then moving on to hyperpolarization of minus 40 to 50 millivolt, and then going back to normal repolarization of minus 70 millivolt. Again, that is an energy process. But here we need another enzyme which will remove the neurotransmitter from postsynaptic neuron. For example, if the neurotransmitter is acetylcholine, then the enzyme will be
Acetylcholinesterase.
cholinesterase, which will be removing those chemicals, because the enzymes have to remove those chemicals to bring that postsynaptic cell back to normal state.
Okay. So here it is what you explained just now.
Yeah.
So we can see the vesicles getting acetylcholine or neurotransmitters,
So
for
in
example.
this case, we will say the gates which are taking the sodium in are ligand control gate. Ligand control gate because they are controlled by attachment of the neurotransmitter. Till neurotransmitter is not attached, the gate cannot open.
Okay, you mean the receptors.
So in the case of the previously the axon, they were ion stimulated or the voltage stimulated gates, but here it is ligand stimulated gates which are there on the dendrites of the postsynaptic neuron. That is the only difference.
How fast can this action potential travel? Do they travel faster in some animals and not in others?
No, it totally depends upon how fast the signal is moving or how fast the stimulus is being given,
Okay.
because that will increase the threshold.
Also, if I am to explain, I would say the current depends on the voltage, that they are directly proportional, but they are
Inversely
inversely
proportional
proportional to the resistance.
so here, who is controlling the resistance? It is a myelin sheath which is lowering down the resistance.
That's exactly shown here.
So what will happen in the case of multiple sclerosis? The resistance cannot be lowered down. Therefore, the message will be, or the transmission will be very, very slow in the patients of multiple sclerosis. Again, here the process is explaining how the action potential is moving all across the nerve fiber, which we discussed previously. So this is which I was saying that the internode is having myelin sheath. So you can see here only nodes of Ranvier are the one which are having the sodium or potassium gates, because it is only this junction where the ions are going to move in or going to move out. So the red place is showing how the first internode has received the action potential and now it is moving to the second. So by the time action potential moves to the second internode.
So what are endorphins?
Endorphins are the neurotransmitters which help us to combat any kind of pain and put us in normal or happy state.
Okay, they make us feel happy, is it?
Yeah.
So when do we release them?
In any of the painful situation.
Or maybe
even while consuming certain stimulants, say
like
chocolate or even tea or coffee.
I like chocolate. Yes. So continuous exercises also release endorphins, and that releases pain, relieves pain, right?
Yeah,
and
keep
makes
us
us
healthy.
Healthy, wealthy, everything. Well, let's talk about vision. For vision, we have various sense organs like the eyes, and we see or rather perceive light with the help of the cornea and the pupil. The light enters through the lens and then comes onto the retina or the film, which is at the back of our eye, as we are able to see here. And that senses and picks up the message in form of through or via the nerve fiber.
Because
The-
rods, they work in the dark, and the cones are the one which perceives the different kinds of light or the colors. So cones are active during the bright daylight and rods are active during darkness.
they are the
Part
the
of retina.
retina. And if you see the small opening, which is the pupil where the light comes in, and of course it crosses first the cornea, the outer covering of the eye, and then via the pupil the light enters into and then goes through the lens. The lens helps in allowing the image to be formed on retina.
Because lens is attached to some here orange color structures which are the ciliary apparatus which keep on changing the focal length depending.
So here is a cross section of the eye. Same eye, you have two chambers. One chamber just outside the lens to the exterior called the anterior chamber, and one the posterior chamber, and both have fluids.
The anterior chamber fluid has aqueous humor which gives nourishment to lens, and the posterior chamber, the vitreous humor, it
Or the
actually
fluid.
maintains the shape of the eye
Okay.
because that jelly-like material.
What is this iris? What does that iris do?
Iris is the one which has a central pupil
Mm-hmm.
which acts as a
Opening.
diaphragm,
Okay.
just like the diaphragm of a camera.
Okay. When it is, we have lots of light outside,
Then the
iris
pupil
would
can constrict under
Okay.
the parasympathetic nervous system
And
action.
what is that choroid? I can see a layer
The
of
choroid is the one which creates the darkness so that the light rays are not reflected out because it is rich in melanin pigment.
Okay. And what about this macula? The small
Macula
dot.
is a region which is also called as yellow spot. It is the only part of the retina which doesn't have rods and it has only cone for the finer details
Okay, the retina has rods,
concentrated towards the posterior side.
and that's what you were talking about. Here we are able to see the rods and
So
cones.
depending upon the shape, the upper part of the rod is cylindrical and the cone is pyramidal shape. See, the cones are of able to perceive three different color, three basic color, that is red, blue and green. So the red color, erythrolobe, chlorolabe and then the cyanolabe, we have three kinds of cone, and mixing of these three basic colors create the rest colors
The rest
for
of the
the
colors.
eye.
So we are able to see the rods are very sensitive to light,
That is, they get bleached in the presence of bright light.
and we are also able to see the rods and cones here.
So
The rods are
help
responsible
us
for vision in dim light.
in the dim light or in darkness. So all those nocturnal animals, for example owl, it has no cones in the eyes because they have to see only at night.
Mm-hmm. So cones detect colors.
Yeah, that is, if we have to see a minor dot on our shirt, we cannot see at night because it will be only the daylight which will help, because cones help us for minor detail.
Mm-hmm. Let's go to the next sensing organ for hearing, that is the ear. It has the outer, middle and inner chambers.
So we will say outer mainly concentrate the sound waves, the middle one is the one which resonate, and the inner one which actually help us for hearing as well as for balancing the body.
Oh, so it's wonderfully explained here through this animation where you're showing the external, the middle ear and the internal ear.
Yeah.
The internal ear is associated with the nerves which pick up the sensations.
And they help us for balancing.
So those sound waves come, strike the external ear.
Till external ear they'll be traveling in ear, and once they strike even the middle ear has three ossicles or the three bones. We call them
The hammer,
yeah.
the anvil and the stirrup, or they are referred also as malleus, incus and stapes.
Exactly.
And once these three small bones and ossicles have vibrated,
Because they are attached to a membrane of cochlea which is highly resonating, So little bit vibration will also be perceived by cochlea,
and will
and
send the action potential
thereby
into.
again the action potential as in the case of the nerves. And that's how the cochlea, the sensory cells of cochlea called as organ of Corti, they will perceive those vibrations, send it to brain, and
Here
brain then perceive it in the form of speech or language.
So here is the cochlea, which is bluish in color. So the as I understand, from the outer ear, the waves come, strike the tympanic membrane, and from the tympanic membrane, the vibrations travel through small bones called the ossicles, the malleus, incus, and the stapes, and then
the vibrations are passing on to semicircular canal, the three tube-like structure in the top blue color. They will be helping only for balancing the body in relation to gravity.
And then the senses will
And the
pass down.
senses will
Have a look at it. Here is the cochlea, which is being cross-sectioned and simplified in a diagram.
So there is a green portion that is called as
endolymph,
yeah, which is rich in the viscous material endolymph.
surrounded by the perilymph,
Yeah, perilymph.
which also has a fluid.
Perilymph has a fluid, but it doesn't have the ear stones or
Okay.
calcium carbonate crystals. They are only in endolymph.
Okay.
They perceive actually because they keep on moving or oscillating. Therefore, they send different vibration to our nerves.
Okay.
That is auditory nerve that perceives those vibration. Take it to brain. Brain has already recorded language or speech, particularly the temporal region.
This is the same figure here, again
Yeah.
shown in a beautiful manner.
So this is how through auditory nerve it will be going to brain from cochlea.
So,
So
it is
the-
a cross section of a cochlea.
So it has three chambers inside: the vestibular canal, the tympanic canal, and the middle one, the cochlear duct. So it is only
In the cochlear duct, I can see small cells. The
yeah,
organ
they are
of
the sensory
Corti.
cell organ of Corti. They are the one actually which perceive the pressure by the ear stone, or there is a tympanic blue color membrane above organ of Corti. And another thing, the vestibular canal and tympanic canal both are filled by perilymph, and in perilymph it is a media where you will not, it is not viscous, it is transparent, and it will not be having ear stone. It is only the cochlear duct which is rich in sensory cell,
Yes.
and which is rich in the ear stone. The roof of cochlear duct is highly resonating, and even the base is highly resonating. Little bit movement in the vestibular canal or tympanic canal can send those vibration to the sensory cells of organ of Corti,
and
which
they
is-
can. And here you
the
are.
detailed structure of the organ of Corti. The blue color are the sensory cell, generally 30,000 to 50,000 in each of the ear, attached to highly viscous membrane, tectorial membrane, which is rich in the ear stones, and their movement or the pressure creates the of the sensory cells.
This is another way of showing it.
That
How
how
the sound waves travels through the
attach the
tympanic
tympanum
membrane,
and then
through
through
the-
ear ossicle, how it moves to
the,
semicircle
the
canal.
oval window,
Yeah.
where it is being attached.
Because that is a region which is highly resonating, thin
Okay.
membrane which is
Then
highly
it bring out those waves. The next one is our nose, smelling organ. So all the chemicals that we pick up through our nose, they travel into this chamber, and on the top you have this olfactory sensory cells.
because there is a thin film of mucus, so that mucus layer picks up the chemicals and then passed on to the sensory cells of our nasal chamber.
So again, do you think we would say these are again the chemical
The
bound-
ligand binding type of gates again, because
Okay.
it is ligand are those chemicals which we are picking from the environment, and they are going to open the sodium gates, and then the same process will be there that set in.
The tongue. We have the sweet taste buds in the tip of it, and then the salty ones around it, and the bitter ones at the back of our tongue, isn't it? So these different senses are being perceived by sensory cells, and we can see it here in more clarified manner.
So these tastes are because of the whole of the tongue is covered by some gustatory receptors, which are very
Sensory
sensitive to the
cells.
taste.
Mhm.
Those sensory cells are of different kind. The
Ah.
most abundant, these are the small ones, largest in number, filiform, Then we have the fungiform, the round ones, which if we see our tongue in the mirror, they will appear as a red spots. So then they are the one which then foliate ones which pick up in the region of the south.
You mean there are many kinds of chemosensory cells,
chemosensory cells
which pick
on
up
our
different. So for touch on our skin or just beneath the skin, we do have many kinds of sensory cells.
And different kind of cells, they pick up different perceptions. Some
For
are
heat,
sensitive to heat or light
cold,
or cold
light, touch,
or pressure.
Okay.
And the deepest placed are the capsulated one, which are attached near the ligament and the muscles. They are called as the strong pressure points. The Pacinian corpuscle, they are capsulated. Rest all are the open free nerve ending.
So what are these Merkel's discs?
Merkel's discs are generally present near the in the vertebral column.
They are
near
superficial,
the disc,
quite
yeah.
superficial.
Quite
and,
superficial. Even Messner's corpuscles on our fingertips are superficial.
And some of the nerve endings are just free. They don't have any corpuscles.
All of them are free. The capsulated ones are the strong pressure point. They are called Pacinian corpuscle.
Okay, then I can see some Krause and bulbs too.
Krause and bulb always pick up the cold sensation.
Okay.
And the Ruffini's free ending always pick up the heat sensation.
Okay.
So
and
both are
what are these
thermoreceptor.
Pacinian corpuscles?
Pacinian corpuscles are the capsulated one, and they pick up any changes in ligaments and muscle. They are the deepest placed.
Let's go to the next system, endocrine system. We have many organs, and these organs, they give off some chemicals. We call them hormones. And these chemicals, they don't travel through what we call tubes or ducts, and they are simply flushed in or put into the bloodstream. And the blood picks these hormones and then pass it on to some other organs for the action. So some organs, they produce hormones, they are put into the blood, and the blood takes it elsewhere for the action.
That means you say that the one which are producing hormones are the endocrine glands, and the other one which are receiving are the target organs.
True. So we have the hypothalamus, the anterior and posterior pituitary gland, the thyroid, the parathyroid, adrenals, pancreas, ovaries, testes, pineal glands, lots of them. So they produce hormones, the chemical messengers which bring about the action. And we always say they are produced at one place, they act in different place, as we called it on the target cells. So most of the hormones are proteins. Of course, there can be others too, like steroidal hormones.
So the difference between the protein and steroid is that steroids can penetrate deep in the cell. They can go enter the cell and then act. Whereas the protein one, they cannot enter the cell, but they have the receptors on the top of the membrane.
As you are able to see it here.
So that means every protein hormone will have a secondary messenger to act, and secondary messenger is a cyclic AMP, amino monophosphate enzyme.
Okay. And
the steroid ones,
they also make use of ATP molecules, do they?
They do, because the ATP breaks down into AMP, and AMP acts as a secondary messenger for all the protein hormones, whereas steroid hormones, they do not have any secondary messenger because they have capacity to pass through. So steroid hormones can penetrate the cell, can directly affect the DNA, whereas the peptide, they cannot penetrate. They need secondary messengers.
And they act on the surface, on the receptors.
On the receptors.
So here is steroidal hormone getting inside.
Yeah, it can directly go and
after getting inside the cell,
it can even penetrate. Its receptor will be inside the cytoplasm.
True.
It will get attached to that. Then the complex can enter through the nuclear pore, directly can go and alter and affect.
And that's what is happening here.
So produce the desired messenger RNA and desired.
Lots and lots of hormones produced by various, I think every one of us must know the action of various hormones produced by various endocrine glands. Here's a chart which is suggesting their actions.
And also their nature.
True. Let's go to the next one. How do we reproduce and how do we develop? So a man and a woman, would produce gametes, what we call the eggs and sperms. They're haploid cells, and they develop through a process called meiosis. And once the sperm and the eggs are being produced, they come, unite by a process called fertilization. And once the fertilization has occurred, it would produce a one-celled structure, and that would be called a zygote, which will be 2n. Of course, the sperm was N, the egg was N, and when they come together, they form 2n structure, the zygote. This repeatedly divides and develops into a small mass of cells called morula, which subsequently develops into an individual, and thus we all develop. Here we are trying to show how by meiosis the eggs and the sperms are being formed. In a meiosis we always see that yes, the 4 cells are formed. In the process of gametogenesis in man, all the 4 cells develop into sperms.
So you mean to say spermatogenesis, their one cell leads to 4 sperms, and in the case of female, in oogenesis, the one cell will produce only one active ovum,
and
the
3
rest
other would form what we call polar bodies.
They will be degenerative.
Degenerative, they would lose themselves, they will be reduced, they won't have much of cytoplasm.
So that means that's the difference between spermatogenesis. In spermatogenesis resultant 4 cell, in oogenesis only one active ovum.
True. Let's talk about female reproductive structure. Of course, we see that there are 2 ovaries in the abdomen. The ovaries of a female are never outside the body. The ovaries are surrounded by oviducal funnels, which leads into what we call the fallopian tubes. The 2 fallopian tubes join together and get into a muscular structure called the uterus, which leads to outside through a tube called the vagina. Well, let's see. Here we see an ovary, and in the ovary we are able to see lots of cells, and each of these cells are surrounded by many more follicular cells, and the cell which is right in the middle is nothing but the egg cell, and the follicular cells keep giving nourishment to the egg cell so as to develop it ultimately.
So
and
all
being
this
released.
process occurs under the influence of the female hormone estrogen.
Okay.
That is helping in development of the ovum.
And once the egg has been released out, the leftover follicular cells, they almost degenerate and form a
this yellow colored
reduced
structure, corpus luteum, which will be secreting the second hormone, progesterone.
Okay, probably releasing the hormone progesterone, thinking that yes, there could be a pregnancy, right?
And if the pregnancy occurs, then progesterone will be retained so that the pregnancy can continue.
Don't you think release of progesterone will stop the process of formation of any further eggs and releasing or ovulation?
Because progesterone is antagonistic to estrogen and will suppress that.
So here is the structure which I talked about. The 2 ovaries surrounded by the oviducal funnels leading into the fallopian tubes, which conjoin into the uterus and ultimately leading out through the vagina. And on the orifice you have the membranous lips called male reproductive structure constitutes the testes, which remain in fine sacs called the scrotum or the scrotal sac outside the body. They need 2 to 3 degree temperature less than the body temperature so as to produce the sperms. So that's the reason why males have the testes in thin sacs outside their body, and these testes have lots of tubes wherein sperms are being produced called seminiferous tubules. They lead out, come out of the testes in form of a coiled structure called epididymis, and then they straighten out via the vas deferens, and thereafter the 2 vasa differentia join at a junction where you have a large gland called prostrate. This supplies lot of materials and nourishes the sperms, and they come out through the urethra, and the urethra at the end is, here is the testis, a cross section wherein we are able to see the seminiferous tubules. They join together to form the what you call the retitestis, and then it leads to what we call the efferent tubules and the head of the epididymis, which is a very coiled structure. And when it straightens out, it is referred to as ductus deferens.
So it is the head.
How the sperms travel. They are nourished by the glands and then ultimately ejected out.
So one ejaculation can have 250 to 500 million?
Oh, lots of sperms. And a man continues to produce sperms every day. And each of the sperm would contain in its head region the DNA, the mitochondria, and various enzymes for penetrating the egg. And here the sperm looks like with one flagellum. When the egg and the sperm they meet, that process is referred to as fertilization.
Which can strictly occur inside the fallopian tubes, nowhere else.
True. The sperm enters and make the egg a zygote, what we call 2N structure. So with lytic enzymes, they dissolve the membrane of the egg and get inside, right?
So that is called acrosomal cap, which
Yes.
has lytic enzymes.
Yes, the enzymes, there are enzymes in the acrosome or the head of the sperm, which helps in dissolving the egg and penetrating. Large number of sperms are being surrounded surrounds the egg.
Only one can enter.
Only one can enter. The moment one enters, it forms a fertilization membrane
So
and
that
allows no more other sperms to get in.
Okay.
And during the course of event, the morula divides repeatedly. The blastomeres, the small cells that are formed, they form the organs. So that's a process called organogenesis, and ultimately you have a fetus. And when the fetus is well developed, it develops all the structures, as you're able to see the eyelids, you're able to see small heart, and it is connected, and it is within a sac called amnion, and it has a fluid called amniotic fluid. So
that is shock absorber?
yes, and it's a nourishment. It provides,
Placenta.
and also it's a protector. Yes. The last one happens to be the skeletal system, if not the least. And here's a man dancing. Seems to be a skeletal dance. Well, we consist. We have lots of bones and cartilage. Do you know how many bones you have in your body?
I think they are more in our childhood.
Yes, and they gradually get fused, and we have only 206 bones in adulthood.
Okay.
It protects. And you know the bones, they have a cavity, and in the cavity you have content what we call the marrow, and that produces your blood and blood cells.
Okay, so that is a hemopoietic tissue that makes.
This is what I have tried to show here.
Okay.
And how many muscles do you have?
No idea.
More than 600 muscles, 639, you can imagine. And the study is referred to as myology. And muscles, some are under our control, some are not, voluntary and involuntary.
So this is the only tissue, muscular tissue, capable of contraction and extension?
Yes, that is the beauty of this tissue. And many are paired and some are unpaired. Can you answer this? Which of these have more number of muscles and how many? Let's see. Head, 53. Oh, wow. And the back, we have 180 muscles. And the legs, which holds the whole body, has 124. You can imagine that. And our muscles produce a lot of energy. Lots. And do you know what is so unique about it? You said that.
Yeah, they can contract and they can extend.
Yes, and they're very elastic, irritable, and they are strong because of their tenacity. Now the question is, how do muscles contract?
I
The
think
nerves.
we learned that
Yes,
before. The
the
end
nerves.
plates, that is, they are the one which are deep in the muscles and they receive signals from the nerves,
The nerves end releases acetylcholine,
and they are the one which are going to then stimulate the muscle cell.
and they're attached. The end bulbs are attached to the muscle fibers
Which
in
are
this
also
manner.
called end
It's 10
plates.
o'clock.
And each of these muscle fibers may have a large number of actually single cells called multiple nucleated cells, and each of these fiber contains myofibrils, in fact, which are made up of contractile small units from one Z line to another called
So you're talking about the striated muscles that
Oh
means.
yes, of course. These have striations and hence called striated muscles. And these sarcomeres are made up of fine, thin filaments and thick filaments. So the thinner ones are referred to as actins, and the thicker ones are called myosins, which alternate one another. Here, you are able to see the thicker ones are right in the middle in red colored. That is myosin. And alternatingly, we have the thinner filaments in between called actins.
So sarcomere is from one Z line to another Z line.
That's true. So from one actin end to another actin end of another end. So that is, and the midline is sometimes referred to as M line, and these actins are held together. Here it is. The thicker ones, the myosin, the thinner ones are the actins.
And this is the combination of the myosin head with the actin through calcium that the A zone gets reduced during contraction.
Yes, and the two actin
A
filaments
zone, or we
from the
will
two
say the
ends.
M zone, the middle zone gets reduced during contraction.
Yes, the two actins which have a gap inside that you're talking about, they get reduced when they contract. So in fact, the myosin and the actins, they slide over each other,
So we will say sarcomere will be contract.
reduce, and that's what is happening here. So one can understand how the actin and the myosin heads, they attach themselves, they swivel, squeeze, slide over another, make use of ATP molecules, and thus contract. In this process, to attach, they need calcium ions. Without that, they cannot contract. This process also requires ATP. That is, this is a good diagram of contracted condition shown here. Here are some of the practice questions. In the next chapter, we'll be doing what is the innate immunity, what is acquired immunity, with various examples. We need to know about the excretory system, functions of kidney, structure of eye, and a nephron. One must have knowledge of the process of muscle contraction, how action potentials are set in, and that's it for now. Thanks to Dr. Aman and my listeners.